people in the cheap seats above. You hear me? Okay. So with this. Okay. Okay, so good morning, everyone. I'd like to start by thanking the organizers for the opportunity to present my work here. As it was said, my name is Iddo. I'm a student in Iddo Weiner lab in Tel-Aviv University, and I'm going to talk today about the evolution of shine-Dalgarno ribosome binding in chloroplasts from a systems biology perspective. So let's see if we, okay, this works nicely. So a quick, a brief overview of how I got to this topic. So in our lab, we mainly work with algae, and a project I've been involved in for some time now was improving our ability to express synthetic genes in the nuclear genome. So what we did, we designed sequence optimization algorithms and other similar methods, and what you can see is that in time this worked. And gradually, and we're able to get higher and higher expression. And for a while, we were very happy with this, until I first tried to express the gene in the chloroplast. And when I did this, my first attempt at chloroplast expression outcompeted everything we've ever achieved in the nucleus, which made us realize that we need to allocate many more resources to understand gene expression in the chloroplast. It's a very interesting organism. So just to quickly remind you, we know that the origin of the chloroplast is a free-living cyanobacterium that over 1 billion years ago was engulfed by a larger eukaryote cell to get the first prototype of a plant cell. Okay, now when the first chloroplast genomes, of course the chloroplast has retained its own genome, and when these genomes became public around 20 or 30 years ago, one of the biggest questions was what are the evolutionary differences between this chloroplast genome and its bacterial ancestral genome. And here are the observations. The early reports show that when you look at the mRNA half-life, you see that in protobacteria, and now I need to say something important. The way that most people tackled this question was to take chloroplast genomes and compare them to the easiest bacteria out there, or the one with the most data, which is just simply E. coli. So in E. coli, the mRNA half-life is typically very short, usually seconds. In chloroplasts, on the other hand, it's much longer. And the second interesting difference was the correlation between mRNA and protein levels. In E. coli, the mRNA levels are a pretty good approximation of the protein abundance. However, in chloroplasts, we see many times that the mRNA fluctuates wildly and the protein abundance is, barely shows any effect. So all these observations together have led to the common dogma in this field that says, okay, in protobacteria, the dominant regulatory step in gene expression is transcription. Of course, this is, in general, it's crude. It's not necessarily true for every single observation, but this is like the general mindset. Whereas in chloroplasts, the translation became much more dominant phase in gene expression. These were the observations. Now, the interesting question is, what is the evolutionary process or the selection that leads to these observations? Simply the question why. And you read, here are some explanations that you read. You find them all around. So many people say this must be close symbiosis with the eukaryotic host, right? The chloroplast genome now needs to be coordinated with the nucleus, coordinated with cell cycle, and this may have to do with the very small size of the chloroplast. It lost many of its genes over the years. Okay, I'm going to leave you with this. Remember it, it's important. And now we're going to move to the main question of my research here. Regarding the shine-Dalgarno mechanism. So just to quickly review this, the shine-Dalgarno mechanism is actually an RNA-RNA interaction between the mRNA and the 16S ribosomal RNA. Okay, it's just simply by base pairing. And this is the canonical model that explains how the bacterial ribosomes find the start codon and translation is initiated. Now, very important for our talk today, you need to remember that this process is spontaneous. It doesn't require ATP, GTP. It's energetically favorable. And now in E. coli, when you take these ribosome binding sites, we can call them, or shine-Dalgarno motifs, and you delete them, what happens is that translation is knocked out. Now in chloroplasts, this happens sometimes, but sometimes it doesn't. Sometimes we delete what looks to us like a shine-Dalgarno sequence and nothing happens. So one of the first things we did, we wanted to look at this, we wanted to look at these questions from a very wide angle. So what you see here for the two groups, chloroplasts and protobacteria, we built a very large database. Each row is a unique organism selected to represent its genus. And what you see here are the 5' EPRs. Now in the pixel color is the sequence conservation. So you can see in protobacteria, the area where we expect to find the shine-Dalgarno motif is very much conserved. However, in chloroplasts, not so much. The pattern is much noisier, which is what you could expect given the empirical results. However, we also created a filter based on a permutation test that shows us that we can differentiate between chloroplast genes that don't have any shine-Dalgarno sequence and genes that have a very strong and clear protobacterial shine-Dalgarno sequence. Now this hypothesis of ours that we kept to ourselves in the lab got a very significant strengthening recently. This nice paper knocks out the shine-Dalgarno mechanism by mutagenesis. I'm not going to go into the details. And what you see is that this is deleterious for plants or plant cells in this case, okay? Almost completely not viable. Meaning the bottom line, which I want you to take from this slide, is that the shine-Dalgarno mechanism is active, still very much functional in chloroplasts for specific genes. Now this raises an important question. If you put together all the pieces I've been talking about, we know that this mechanism is rapid and spontaneous. We know it's suitable for gene expression regimes where transcription is dominantly rate limiting, right? As soon as you have some mRNA, you want all the ribosomes to immediately jump on it, get as much protein as you can before it degrades. But I just told you that translational regulation is more prevalent in chloroplasts, so we expect to see some kind of additional layer of regulation that lowers the spontaneity of the shine-Dalgarno mechanism and acclimates it to the chloroplast gene expression environment. So the first thing we suspected is that maybe the anti-shine-Dalgarno, okay, the sequence on the 16S has been mutated during evolution, and very quickly we've seen that this is not the case. This sequence is very much conserved throughout our database. So the next thing we did, we examined the PDB structures, and we extracted the entire 16S 3' edge, the whole piece that sticks out from the ribosome mass. And what you can see here very nicely are these two positions with high variability between protobacteria and chloroplasts. Now, what we thought is that maybe these mutations have to do with the structure of the complex. So we simulated the secondary structure of all these RNAs. This is the structure in bacteria. You can see this nice AGG hairpin loop. And here are the anti-shine-Dalgarno nucleotides. However, in chloroplasts, we get something completely different. Sorry, the hairpin loop is gone, and now the anti-shine-Dalgarno nucleotides are caught up in base pair interaction, making them less available to form the spontaneous interaction with the mRNA. We wanted to test this hypothesis empirically, so we built two vectors for chloroplast expression in Chlamydomonas. Both have a reporter gene with a nice shine-Dalgarno sequence situated upstream of the start codon. Vector A has the native 16S, which folds like this, and vector B has this slightly modified version that folds like this, like an E. coli, and we expect higher expression from vector B. So I'm gonna go through this quickly. We did this with two different reporter genes. One was a fluorescent protein named Cherry. The other was our hydrogenase that we like in our lab that catalyzes the formation of gaseous hydrogen. In both cases, we see that the expression from vector B is roughly twofold higher than that that we receive in vector A, which very nicely shows that validates our model. Now, to me, the interesting question is, what is the origin of these features, okay? And I'll remind you that when we do this comparison between chloroplasts and proteobacteria, we're actually missing something, right? We need to throw the cyanobacteria into this picture as well, because we know the relations in this family. Now, if you look at this, you see that already in the cyanobacterial group, the shine-Dalgarno conservation pattern is missing, okay? And when you throw them into this alignment, you see that these two regions are variable here as well. And most importantly, when you simulate the folding in cyanobacteria, you get the almost identical structure to what you get in chloroplasts. So if we take all this together, and as we know that the shine-Dalgarno is a hallmark for transcriptional regulation, and lowering its spontaneity, as we see in chloroplasts and cyanobacteria, is we can look at this as a way to examine shift to translational regulation. Then this actually promotes the idea that all this action, okay, all this move towards a more translational regulated gene expression regime actually happened much before endosymbiosis. So we can take all these hypotheses which differentiate chloroplasts from bacteria, okay, and I'll say gently put them aside. Okay, and instead we need to look for reasons, for evolutionary reasons that differentiate chloroplasts and cyanobacteria together from proteobacteria. These will be completely different reasons. I'm just laying a few of them. This could be adaptation to oxygenic photosynthesis. This could have to do with the fluctuations in light intensity, and this could have to do with slower growth rates. With this, I'd like to end. There are some important thank you messages I want to say. So first of all, to my supervisors, Iftach and Tamir, and all the friends from the lab. Special thanks to Noam, who was a full collaborator of mine in this work, to the university, and to you for listening. Thank you very much.